Hi there, my name is Anthony. I'm heading into my third year B.Sc, and today I'll be talking to you about neuron morphology of burst firing and regular firing neurons in the mouse medial prefrontal cortex. Essentially, this is a really interesting region of the brain to study because it governs higher order cognitive functions like attention and decision making. Acetylcholine, a neurotransmitter, acts in this brain region to govern these cognitive functions, and more specifically, it acts on two main nicotinic acetylcholine receptor subtypes predominant in this region, which are alpha-7 receptors and alpha-4 beta-2 receptors. Both of these receptors are present in the medial prefrontal cortex, but what's interesting is neurons with the alpha-7 receptors are associated with burst-firing neurons, whereas neurons with the alpha-4 beta-2s are associated with regular firing neurons. And what's really cool is that when you apply acetylcholine to neurons with the alpha-7 receptors, and you measure the cell current over the cell membrane, we see that the neurons with the alpha-7 receptors have a very sharp, rapid response, whereas neurons with the alpha-4 beta-2 receptors have a much more slow and broad response. So the purpose of this study is to compare the morphology versus function between burst-firing neurons with alpha-7 receptors and regular firing neurons with alpha-4 beta-2 receptors. We're also going to look to compare the sex differences between burst-firing neurons with alpha-7 receptors. And to do this, we extract thin brain slices from CD1 mice, and essentially we're going to categorize what type of receptor a neuron has by measuring their response to acetylcholine doing whole cell electrophysiology experiments. At the same time, we can inject that neuron with biocytin and visualize it under bright field microscopy, and then we can reconstruct that neuron in 3D and conduct a Shoal analysis, which is essentially concentric spheres around the cell body in 25 micron increments that measure properties like length, diameter, and volume, which give rise to these Shoal graphs. And so before I get into my data, I'm just going to orient you with a Shoal graph. 0 indicates the cell body, and to the right indicates the apical dendrite, which is the main dendrite of a neuron. To the left of the 0 here indicates the basal dendrites, which is every other protruding dendrite from the cell body. And again, we're measuring properties in 25 micron increments from the cell body, which include length, diameter, and volume measurements. And so what we see here is that burst-firing neurons indicated in the blue compared to regular firing neurons indicated in the black, show that for its basal dendrites, greater basal length, greater basal diameter, and greater basal volume, whereas for the apical dendrite, there's less apical length, less apical diameter, and the apical volume did not significantly differ. And this is reflected by the traces here, where the apical dendrite of burst-firing neurons is clearly less complex than the regular firing neurons, whereas its basal dendrites are more complex than regular firing neurons. And this is really interesting and significant because dendrites are the main receivers of signals from other neurons, and this may play a role into their function in the medial prefrontal cortex. Furthermore, we actually see sex differences as well. Here the males are indicated in blue, the females are indicated in pink or red, and what we see is that males have greater basal length, basal diameter, but less basal volume, whereas they have less apical length, less apical diameter, and less apical volume. And what you can see that male neurons are clearly less extensive than their female neuron counterparts. And I want to note that these neurons are taken from mice aged 15 days old to 20 days old. And so this may actually outline a developmental difference in burst-firing neurons between males and females. And so to conclude, burst-firing neurons with alpha-7 receptors exhibit larger basal and smaller apical dendrite trees. Regular firing neurons with alpha-4 beta-2 receptors exhibit smaller basal and larger apical dendrite trees. And lastly, male burst-firing neurons with alpha-7 receptors exhibit smaller basal and smaller apical dendrite trees. In the future, we're going to continue to investigate these effects across different age and sex groups. We're also going to look to measure dendrite, spine density, and morphology. And lastly, we're going to assess the relationship between receptor function and neuron morphology, the strength of receptor function and neuron morphology. Thank you so much for listening. I hope you enjoyed, and have a great summer. Thank you.